today's edition of biology. We're still continuing with the chapter digestive system, and right now we are about to finish. We are approaching the end of the chapter. So today let us discuss about enzymes, and of course we'll talk about their characteristics and the fate of the end products of digestion, the likes of glucose, amino acids, and fatty acids and glycerols. So first of all, let us define an enzyme. So enzymes are biological catalysts, of which takhala kunena zimenezi for a long time now. So enzymes are biological catalysts, meaning to say that these are chemicals or things that helps to fasten the chemical reaction in our bodies. So that is an enzyme. So we have characteristics of enzymes. One, they are proteins in nature. Enzymes are proteins in nature. Some of you, we did this in class. I think we don't have to waste much of our time here, since for some of you, yes, you already have this information. They work at optimum temperature, which is 37 degrees Celsius. It means these enzymes nthawi imene amagwira bwino ntchito ndi pa 37 degrees Celsius. So when the temperature is very low, they are inactive, and when the temperature is very high, enzymes are denatured. So this is what happens. So they are inactive when the temperature is very low and denatured when the temperature is very high. They work better at optimum pH. Optimum pH, we are looking at acidic condition and alkaline condition. Tanthozonera kuti ma enzymes ena amagwira bwino ntchito pamene pali acid, ena pamene pali alkaline condition. Chitsanzo, in our stomach imapezeka hydrochloric acid. So pepsin, which is an enzyme that digests proteins to polypeptides, it works better under those conditions. As a result, zimapangisa kunena kuti enzyme imene chifoti imagwira ntchito bwino pamene pali acid and mimba muli hydrochloric acid, the acid helps this enzyme to work better because it works better under acidic conditions. And another good example is trypsin, which is found in the small intestines. Trypsin amapezeka mu small intestines, whereby many mumuti nkakamba zonena kuti muli alkaline condition imene imabwera from the chemical imene timatchula kuti sodium hydrogen carbonate, which is an alkaline condition. Then we also have another characteristic. They are specific. It means enzyme imene ikugwira ntchito mkamwa singapezekenso m'mimba. Komanso enzyme imene ikupanga digest ma proteins singapange digest carbohydrates. It means they are specific. Enzyme yonse ndi ntchito yake special. They are catalyst. Catalyst, it means they speed up chemical reactions. That's they are catalyst. Then let's go to the fate of the end products of digestion. What happens to the end products of digestion? Ndiye tinaona kunena kuti starch kapena carbohydrates, they are converted to glucose. Ma proteins amakhala ma amino acids, ma fats amakhala ma fatty acids komanso glycerol tinaona zimene zicha. So tiyambe ndi the fate of glucose. So glucose is converted to glycogen by the liver for future use, while the right amount of it is sent to all body cells by the blood, where it is used for respiration. Now glucose akakhala kuti alipo ambiri m'magazi mwathumu, liver imapanga convert to glycogen. Tikapita ku endocrine system kuja timapeza kuti kumene kucha kumapezeka insulin amene amachokera ku, of course insulin amachokera ku pancreas, imene ndi enzyme imene imazagwira ntchito pachani pa glucose kuzabalansa levels of sugar in our body. So glucose amasanduka glycogen nkusungidwa mu liver, oyina amapangidwa released pang'ono kumakagwira ntchito ya respiration. Remember, glucose sugar ndamene amathandizira kuti tizikhala ndi energy. That is why tikazaona mapatient ambiri akakhala foka samadya amayika pa drip ya glucose, though amangooneka ngati madzi koma pali drip ina imakhala ya glucose, not water, because sugar uchi amapereka energy. So it's the same here, kuti glucose amene kupita magazi ntchito yake is for respiration. And when the level of glucose is low in the body cells, the liver converts the stored glycogen to glucose for respiration. Ndekuti glucose uja akachepa, liver imapanga so convert glycogen uja anapanga glucose anasanduka glycogen uja amazapangidwa so reversed to glucose kuti agwire ntchito for respiration. Amino acids. So amino acids are deaminated by the liver. So we have to understand what deamination is. So deamination is the removal of amino groups from amino acid molecules by the liver. It means that the amino groups amakhala kunena kuti it is being changed or removed completely from amino acid. Ndikuti akuchosa kamauko kuti aminoko zimene zikusonyeza kunena kuti ma amino acid iwa amakhala kunena kuti tikamati deamination amakhala kuti protein yasintha pajapa yasanduka amino acid. So deamination is like we are also reversing ma amino acids aja, we are reversing them back to proteins. Tikuona iti. So imene process imene pamakoti amino group ikuchoka from amino acid molecules is what we call deamination. Now we have uses of amino acids in the body. So some forms proteins more, that is why they are reversed to proteins. So some forms proteins to form parts of body cells like mitochondria, ribosomes and chromosomes. Ndikuti tikupeza kuti ma proteins ma amino acid ntchito yamba ndikuti amakhala so ma proteins. Ma proteins aja amagwira ntchito kukapanga form body cells. Kaseo kachati maphunzira kuyambirira kacha kali ndi mapatsa ina mitochondria wa ma chromosomes nditi zitupeti mazipeza mu DNA, things like those. Some resemble themselves to form blood proteins like fibrinogen. Fibrinogen works in a blood clotting. Antibodies, so these are ma cells they fight against infections. Also we have enzymes, they are biological catalysts. So tipeza kunena kuti zinthu zimenezi ndi zimene ma amino acids amapanga akakhala mthupi mwathu. That is about amino acids. In the last part we have fatty acids and glycerols. So remember amene timayang'ana virus timanena kuti lactic ntchito yake makoti ma molecules amene ndakuluakulu azipanga diffuse through lactic. So ma amino acids and glycerols are they diffuse into the villi by the lacteals and transported to the liver by the lymph vessels. You have to understand this. Ma amino acids saenda magazi, they are transported by the lymph vessels. Chifukwa poyamba wachitinena kuti ma amino acids salowa mu bloodstream because they are very large. Therefore, they still need to be transported to the liver. For that reason, they are transported by the lymph vessels. So excess fatty acids and glycerols ndekuti ma excess ndikuti zotsala are stored under the skin, while others are chemically changed to be used for respiration. It means tikati chemically changed to be used for respiration, it means some fats are changed to carbohydrates to be used for respiration, kapena to produce or release energy. That is about the fate of fatty acids and the glycerols. And that is the end of our discussion today. And if you have questions, yes, you may list the questions below. Thank you very much for being there. I think in the next lesson we are going to finish digestive system and we'll dive into circulatory system, which will talk more about the movement of blood in our bodies.